Now this will be a simplified version. It will not explain everything, but will give you an excellent overview of how your body works and how every time you consume genetically modified foods, you are damaging your DNA. Very simply, DNA is a language, and to be more specific, DNA is a programming language, and the order of that language determines the output you get. So you have these two base pairs, as they call it, AT and CG, and that's a very simplified version of the language, and the way it's ordered is going to determine everything about the way you are. This is your DNA. You can see this is a double helix shape and is a perfect antenna, something that I will not get into in this video. However, if you take a look, you can see those two base pairs, AT and CG, basically going through and creating their own language. And that is very specific. If anything is disrupted along the way, it will modify and distort the output. With genetically modified food, or GMO, genetically modified organisms, everything has changed. What they're doing is modifying this DNA structure, the AT and CG, and they are inserting that with a gene gun, that's actually their term, and splicing in what they choose. They may choose a bacteria, they may choose genes from another animal, another species, and another kingdom, and forcefully inject it into this DNA. Of course, this is unnatural. Things from other kingdoms and other species are not designed to be fitted together. That's the way it's supposed to happen, so that everything stays the way it is. However, GMO is changing that.
Alright, so I'm excited today. We actually get to show you the gene gun in action. So stepping back a second, the gene gun is essentially a tool that allows us to take and physically fire DNA into a cell, allowing it to be transformed. It's a slightly different method than, say, agrobacteria or a PEG protocol or something like that. So here's the machine here. I'm going to walk you through the steps basically of how you kind of set it up, and then we'll demonstrate it firing. Little things are. So these little plastic pieces here. So these are 650 rupture discs, which means that they will break at 650 psi. Put that in here, like that. And then there's a little thing up here that you screw. Next, we need our DNA-coded nanoparticles. So that's what we've got here. So if you take a look at here, these are some tungsten nanoparticles that I've already pre-coded with DNA. At some point, we'll probably take you through that step as well, but not today. So what we do is we load that into this little metal piece here. Careful not to scrape the DNA off, because that would be bad. And then just push it in. Make sure it's in there nice and tight. Alright. Got that. And so now we'll put this piece in here. I'm putting it upside down because the shock wave will go down, and that's where we're firing the DNA. Alright. We then need a nice little stopping screen. You don't want the actual membrane going into whatever tissue you're bombarding. And then finally, that means we're ready for the tissue. So here is just an onion peel. Literally just an onion that we bought from the grocery store, and then kind of cut it out, and then took a peel from the inside. It's useful to kind of test run gene guns because, one, it's kind of a flat layer of cells. You can kind of test to see what the pattern is that you're bombarding. 2, they're clear, so you can kind of see things much easier. And then 3, onion cells are pretty decent size, so you can see them fairly easily under a microscope. So we'll put that in here. And so what we've got here is we've got the rupture disc up here that'll designed to break at a very specific pressure. Our DNA coded nanoparticles are here. We've got the stop screen, and then down here we've got our onion cells.
What they're doing is modifying this DNA structure, the AT and CG, and they are inserting that with a gene gun, that's actually their term, and splicing in what they choose. They may choose a bacteria, they may choose genes from another animal, another species, and another kingdom, and forcefully inject it into this DNA. Of course, this is unnatural. Things from other kingdoms and other species are not designed to be fitted together. That's the way it's supposed to happen, so that everything stays the way it is. However, GMO is changing that forever. So take a look at this diagram that I made up. You can see that I just used these letters X and Y are now modifying that sequence to mean something else, but they don't see it like that. They're taking this X and Y from another species of another kingdom that has the trait they're looking for, and then they insert it. Where it goes, they're not sure, because it's a gene gun. That's the way it works. So the effects of which are untold. I'll get to some of those later on. In this diagram here, you'll see that modified piece of DNA being inserted into crops, and this gets put on the store shelves, primarily found in processed foods, but not necessarily just the processed foods. GMO crops are found everywhere now.
Hi, welcome to the Plant Transformation Core Research Facility here at the University of Nebraska-Lincoln. We have the ability to transform all the major crops of Nebraska. That would include corn, wheat, sorghum, and soybeans. In addition, we can transform the model crops like tobacco and Arabidopsis. So what happens here is the researcher brings his gene, or we can synthesize the gene in the lab. We put it into the desired plant species, and then give it back to the researcher for further analysis or do the analysis.
So what we're going to look at today is how we genetically engineer soybeans to introduce new traits. This big apparatus here is called a hood. And what this hood does is it pulls the air through a HEPA filter and blows it out towards me, creating a sterile environment in which to work. To start our transformation process, we use agrobacteria to transform our soybeans. Agrobacteria is a soil bacteria that's found in nature and was discovered in about the 1900s to have a really unique ability. This bacteria has the ability to insert its DNA into plant cells and to modify the plant so it makes food for the bacteria to continue to grow.
And so what scientists discovered that they could do is that they could replace those genes that made food for the bacteria and put their own genes in for whatever they wanted to do in the new plant.
I have some seeds here. I placed these sterile seeds on these plates containing media which contains all the elements necessary for its growth. After 5 days, this is what the germinating seeds look like. Now I'm going to make 7 to 12 horizontal slices. So here I have a bunch of wounded cotyledons, and then here's the agrobacteria that has been resuspended in our co-culture media, and I'm going to pour it over the cotyledons and allow them to sit for about 30 minutes. The bacteria has started to attach to the cotyledons. Now the agrobacteria is attracted by compounds that are generated from wounded plant cells. Remove the cotyledons, wounded side down, in contact with the filter paper. The filter paper contains the co-culture media. And we allow these cotyledons to remain on these co-culture plates for 3 days. During those 3 days, the agrobacteria is going to do its thing. It's going to insert its little piece of DNA into the plant cell. After those 3 days of co-culture has passed, we now move these cotyledons to our selection plates. This media contains a compound, a chemical that would normally kill plant cells. We've introduced into these cotyledons with our agrobacteria a new gene that will allow it to grow despite the chemical that would normally kill it. Also in this media is a compound or a hormone that will allow a single cell to grow into a whole new plant. If the single cell contains that new DNA, all the cells in the new plant will contain that new DNA. After 2 weeks, you can see what has happened to the cotyledons. We've started to see a cluster of new shoots being produced. The hormone, or the plant growth regulator, in this media has altered the normal course of development of the cotyledon. So instead of producing a single big plant, we produced a cluster of buds. You can see here that we have many new plants or new shoots growing from the single cotyledon. And eventually, you will see the start of new shoots growing out of this cluster of shoots. These shoots are our potential transformants. Because the gene gets inserted randomly into the plant genome, and we have no control as of yet where it goes, it could be inserted into a spot that is bad. It could affect the yield of the plant. It could affect the growth and development of the plant. And so we need to make multiple plants from different transgenic events to pick the ones where we don't have any effect on yield or plant growth and development. So one of the ways that we screen for the transformed plant, we look for the presence of the herbicide resistance gene. Now I'm going to apply this to the soybean leaf. I'm going to apply the herbicide. Now in 5 to 7 days after application, if the plant does not contain the herbicide gene, the leaf will look like this. Okay? If the plant does contain the herbicide gene, it will look like this. You can see there's no damage. So this plant contains the new gene, the selectable marker gene, the bar gene, and also the gene of interest. This gene does not. What you see in the greenhouse is not necessarily what you see in field conditions. And field conditions is where you want to know what the plant is going to do.
GMO crops are found everywhere now, so you begin to consume them. You then will digest them, and in that process of digestion, there are little villi, which is like little hairs on the inside of your intestines, and they will absorb that DNA, that information, into your bloodstream.
In our body, absorption of food mainly takes place in the small intestine. The inner wall of the small intestine is highly folded and has many small projections called villi. The structure of the villi is specially adapted for food absorption. The villi are finger-like and large in number. These features greatly increase the surface area for absorption. Moreover, the epithelium of the villi is only 1 cell thick. This provides a very short distance for diffusion of food molecules into the blood. The cell membrane of the epithelial cells has a large number of microvilli. This further increases the surface area for absorption. Each villus has a lymph vessel called lacteal in the center. The lacteal is surrounded by a network of capillaries. These allow the absorbed food molecules to be carried away rapidly. Therefore, a steep concentration gradient of food molecules can be kept across the wall of the small intestine to increase the rate of diffusion of food molecules.
GMO crops are found everywhere now, so you begin to consume them. You then will digest them, and in that process of digestion, there are little villi, which is like little hairs on the inside of your intestines, and they will absorb that DNA, that information, into your bloodstream. When you take in damaged DNA, that results in damaged DNA in you. So in effect, the programming language that you are made up of is being disrupted. Let's go over a few possible effects that occur because of damaged DNA. Diseases, faster aging, organ failure, lowered immunity, and most importantly, something that I talk about consistently, and that is passing down that damaged DNA to future.